Imagine taking a groundbreaking scan of an Alzheimer's patient's brain.
Right.
You've identified this really subtle structural change, something that could completely revolutionize early intervention. But then imagine that entire discovery just vanishes.
Yeah, just completely invalidated.
Exactly, invalidated just because a researcher in another lab tried to analyze that exact same data on a Mac instead of, a Linux cluster.
I mean, it sounds like hyperbole, right? But that scenario is actually the dark secret of modern neuroscience. We are living through this massive reproducibility crisis.
And that is exactly what we are unpacking today. Welcome to the Deep Dive. We are looking at a brand new source today, the NeuroClaw Technical Report.
It's a fascinating paper.
It really is. Yeah. And our mission today is to understand how this specific closed-loop agentic AI system is stepping in to fix this reproducibility crisis in brain science.
Yeah, acting less like a simple chatbot and more like an entire highly coordinated research lab.
Right. So for you listening, whether you are a data scientist building biomedical pipelines, a medical professional, or just, insanely curious about the future of AI, you really need to grasp this shift.
Definitely. Because we're watching AI move past just answering questions into actually conducting auditable, reproducible science.
So to appreciate how NeuroClaw achieves that, I feel like we have to look at the massive gap between what general-purpose AI can do and what neuroimaging actually demands.
Yeah, that's the perfect place to start because we've seen models like Biomni or Origene do incredible things in biology.
Right, like reading thousands of papers to propose a novel drug target for some rare disease.
Exactly. They excel at hypothesis generation. But if you throw raw neuroimaging data at them, like the high-dimensional multimodal outputs from fMRI, structural MRI, or diffusion MRI, they just completely fall apart.
I was actually looking at that section of the report, and the roadblock seems to be dataset semantics.
Yes, the semantics are huge.
Because general AI systems are trained on human language and standard code. But in neuroscience, data isn't just, a folder of images. It's governed by beads.
Right, the brain imaging data structure. That is the crux of it right there. Beads is this highly rigid, really strict organizational standard.
So it's not flexible at all.
Not even a little bit. It dictates exactly how metadata, file names, and directory trees must be structured so that complex neuroimaging software can even begin to read the files.
Okay, and general AI agents just don't natively respect or understand these metadata rules.
Exactly. I mean, they might be able to write an elegant Python script, sure, but they completely lack the domain-specific spatial and semantic awareness required to navigate a BEADS-compliant data set.
Okay, let's unpack this because I want to contextualize what a failure actually looks like here. If an AI misinterprets a tiny metadata detail early on, let's say it swaps the orientation header, thinking a scan was taken left to right instead of right to left, it doesn't necessarily throw an error and crash the program, right?
Not at all. That's the scary part. The script just keeps running quietly.
Oh, wow. So it just ignores the mistake.
Well, the flipped orientation gets baked into the spatial normalization, which then gets fed into the statistical modeling.
So the error cascades.
Yes, it silently cascades through a massive multi-stage pipeline, and then weeks of computation later, it spits out a completely invalid scientific result at the very end.
And you wouldn't even know.
Right. The entire study is ruined, and you might not even realize it until, a peer reviewer spots an anomaly months down the line.
So using a general AI for neuroimaging is basically like hiring this brilliant executive chef who knows a million recipes.
I like this analogy.
Right, but they walk into the kitchen, they don't know how to turn on the specialized ovens, and they don't recognize the ingredients because they aren't pre-chopped. So they grab salt instead of sugar because the bins weren't clearly labeled.
Exactly. They execute the recipe perfectly. The cake rises. It looks beautiful on the plate. But you don't know it's completely ruined until someone takes a bite at the party.
That is wild.
What's fascinating here is that general models just lack the specific domain constraints to realize they are holding the salt. NeuroClaw, on the other hand, was engineered from the ground up to handle the messy, raw ingredients of the neuroimaging kitchen.
Which brings us to its first major capability outlined in the report, the raw data compatibility.
Yes, this is a huge bottleneck in traditional research.
Right, because when researchers get data straight off a hospital scanner, it's usually in DICOM format, right?
Yeah, and DICOM is notoriously bloated. It's packed with patient metadata, scanner manufacturer specs, slice thickness protocols, things that are crucial for the hospital, but an absolute nightmare for machine learning models.
So a human researcher usually has to spend hours, sometimes days, manually sanitizing those DICOM files.
Hours, days, sometimes weeks if it's a big data set. They have to convert them into NIFTI format.
And NIFTI is the standard for analysis.
Exactly. NIFTI strips away all that hospital bloat and provides a clean 3D volume of the brain that research algorithms can actually compute.
And NeuroClaw just bypasses that human bottleneck entirely.
Completely. It has the native capacity to look at a raw folder of messy DICOM files, understand the modalities present, perform the conversion to NIFTI, and structure the output into perfect beads compliance automatically.
Okay, so it does the prep work. That's a massive time saver. But the source material spends even more time detailing NeuroClaw's harness engineering.
Oh, the harness engineering is where it really shines.
Right, this is where it manages the actual computational environment. Like, it's spinning up Docker containers, locking down specific Python environments, and orchestrating complex toolchains like FSL, FreeSurfer, and fMRI prep.
Yeah, and FSL and FreeSurfer are foundational to neuroscience. If you want to strip the skull out of a 3D image or map the topological folds of the cortex, you use those suites.
But I've heard they are notoriously brittle.
Incredibly brittle.
Well, hold on, let me play devil's advocate here. When I think of frontier AI, I'm thinking of generative design or solving protein folding. But half of this report is focused on managing Docker containers and pinning Python dependencies.
Right, it sounds a bit mundane.
Yeah, isn't standardizing files and managing software dependencies just boring IT work? Why are we wasting advanced, highly capable AI on doing a sysadmin's job?
Because treating environment management as just boring IT work is exactly why we have a reproducibility crisis in the first place.
Oh, really?
Yes, the environment itself is an active variable in the experiment. Let's go back to your opening example.
The Mac versus Linux thing.
Exactly. If researcher A runs FreeSurfer on a Mac using Python 3.8, and researcher B runs the exact same raw data through FreeSurfer on a Linux cluster using Python 3.0, the underlying math libraries might round floating point numbers differently.
Wait, just basic rounding differences. And that tiny drift compounds into entirely different brain volume measurements.
Precisely. That tiny drift invalidates the replication of the study.
That is genuinely terrifying for the field.
It really is. So NeuroClaw attacks this by taking total authoritative control of the environment. It doesn't just write the code, it builds the exact virtual laboratory the code runs in.
So it's controlling the variables from the ground up.
Right. It utilizes checkpointed execution, meaning it saves the state of the data at every single intermediate step. And it runs missing file detection before initiating the next phase of a pipeline.
The report also highlights its use of structured audit logs, specifically outputting in JSON format.
Yes, the JSON logs are critical. Every decision, every package version, every single file path change is logged in a machine-readable format.
So it's leaving a paper trail.
A perfect, auditable paper trail. It enforces deterministic cross-environment reproducibility. It isn't just arriving at a scientific conclusion, it is proving its mathematical and environmental work at every single step along the way.
Okay, so it can build a perfect virtual lab, and it can prep the raw data. But we've both seen standard large language models forget the beginning of a prompt by the time they reach the end.
Oh, the context window amnesia, yeah.
Right. So how does NeuroClaw hold a massive multi-day fMRI pipeline in its working memory without hallucinating or just completely losing the plot?
It survives because it doesn't rely on a single monolithic brain trying to process the entire pipeline simultaneously.
So it's split up.
Yes, NeuroClaw utilizes a hierarchical skill architecture. It basically fractures the cognitive load across 3 distinct layers.
So break those down because the architecture is fascinating. At the top, you have the interface layer.
Right, the interface.
From what I gathered, this is essentially the translator. It takes the high-level human intent, like, run a functional connectivity analysis on this Alzheimer's cohort and maps out the broad blueprint.
Correct. The interface layer doesn't know how to actually run the code. It just knows how to plan the strategy.
And then below that, you have the subagent layer. This is where the domain-specific expertise lives, right?
Exactly. The developers gave NeuroClaw 4 very distinct types of subagent skills.
I'm looking at those. You have tool skills, which understand the syntax of external software like FreeSurfer or FSL.
Right.
You have modality skills, which dictate the specific differing pipelines for structural MRI versus diffusion MRI. You have model skills for executing the statistical math.
And the 4th one is the dataset skills.
Yeah. Is it fair to say the dataset skill is basically an encyclopedia for specific brain cohorts?
That is a really great way to frame it. Datasets like ADNI, which tracks Alzheimer's progression, or the HCP young adult dataset, or the UK Biobank.
They all have their own quirks, I imagine.
Exactly. They have unique scanning parameters, weird naming conventions. The dataset subagent is specifically tuned to navigate the eccentricities of those famous cohorts without needing to be prompted from scratch every single time.
And then below all of those subagents, we have the base layer.
The workers.
Right, the execution tier that handles the atomic operations, the actual Python execution, the bash scripting, the file movements.
It's the hands of the operation.
Here's where it gets really interesting, though. It's structured exactly like a construction company. The interface is the architect talking to the client. The subagents are the foremen managing the plumbers and electricians, and the base layer is the worker actually swinging the hammer.
That's a perfect analogy. And structurally, the entire architecture is built as a DAG, a directed acyclic graph.
Okay, so a directed acyclic graph. That basically means information can only flow one way, right? Downhill.
Exactly. You can't have loops. A hallucination or an error at the base layer can't magically swim back up the waterfall and rewrite the original raw data protocols at the interface layer.
But doesn't giving the AI all these separate skills and layers make it slower or cause it to hallucinate more? Like too many cooks in the kitchen.
If we connect this to the bigger picture, it's actually the exact opposite. The DAG structure prevents hallucinations.
How so?
Well, in a standard model, when you feed it a massive undifferentiated prompt containing an entire neural imaging pipeline, the model suffers from attention dilution.
Because it's trying to hold too much in its head at once.
Right. It has to keep the file paths, the biological theory, and the Python syntax all in its active context window at the same time. The model gets confused and hallucinates code that doesn't match the data.
But the terrorist tools of the DAG prevent that.
Completely. By breaking the massive pipeline into these compact, highly defined nodes on the graph, the NeuroClaw agent only retrieves the specific context it needs for that exact microtask.
Oh, I see.
So the sub-agent running the skull stripping tool doesn't need to have the statistical modeling theory loaded into its context window. This intense focus drastically reduces model confusion.
And it also has to be vastly more efficient with token consumption, right? Since you aren't constantly feeding it the entire history of the project at every step.
Yes, it isolates the context. It's highly efficient modular reasoning.
The theory is incredibly sound. I mean, the architecture makes perfect sense. But in the biomedical sciences, a theoretical framework is cheap.
Oh, absolutely. You have to prove it.
Right. You have to benchmark it. You have to prove it actually yields better science. And that leads us to the testing ground, which they call NeuroBench.
The developers knew they couldn't just say, trust us, it works. So they constructed NeuroBench, which is this rigorous system-level benchmark consisting of 100 highly complex tasks manually designed by experienced neuroscientists.
We're talking about tasks like taking raw HCP data, executing fMRI prep, extracting specific cortical volumes, and running a group-level statistical contrast.
Very complex stuff. And they didn't just test one model against this benchmark. They took 10 of the most powerful frontier AI models available.
Like Claude 4.6, GPT 5.4, Gemini 3, Grok, DeepSeek.
Yeah, the heavy hitters.
Yep.
And they tested them in two states. First, naked, meaning the models just operated as standard general AIs. Then they tested them with skills, meaning they embedded those same models inside the NeuroClaw DAG framework.
To evaluate them fairly, they judged the outputs on a strict 1 to 10 scale across 3 specific dimensions, right?
Yes. P, for planning completeness, does the model actually know how to logically sequence the pipeline? R, for tool usage, reasonableness, does it avoid rookie mistakes like trying to apply a functional MRI smoothing tool to a structural MRI scan?
And C, for code correctness, meaning does the generated Python or Bash script actually compile and execute without throwing syntax errors?
Exactly. And one detail I found crucial was their choice of judge.
Right. They didn't have human neuroscientists manually grade all these outputs, but they also didn't let the models grade themselves.
No, that would introduce huge bias. They used a fixed GPT 5.4 model as an impartial referee.
That is vital for benchmark integrity. If you have Claude grading Claude's output, you introduce severe self-preference bias.
Exactly. By using a frozen, highly capable model like GPT 5.4 strictly as the evaluator across all runs, you standardize the grading of those P, R, and C dimensions. It gives you a clean baseline to measure actual capability gains.
And the data from Figure 3 and Table 1 in the report is just staggering. Every single one of the 10 frontier models saw consistent, substantial score improvements when they were allowed to operate inside NeuroClaw.
The DAG architecture literally made every model a better neuroscientist.
The absolute top performer in the benchmark was Claude Opus 4.6, right?
Yeah.
Which achieved an overall score of 72.9% when equipped with the NeuroClaw skills.
It did. But the most dramatic delta, the biggest actual leap in capability, came from Minimax M2.7.
Oh yeah, the absolute jump on that one was wild.
When it was running naked, it really struggled with the domain complexity. But the moment it was placed inside NeuroClaw, it saw a massive absolute jump of 12.97 points.
Almost 13 points.
Yeah.
That proves that the framework itself elevates the underlying intelligence.
Completely. You take a mid-tier reasoning engine, give it a rigorous structure and a flawless lab environment, and it starts producing high-level, executable science.
It really reshapes how we view model utility. You don't necessarily need a trillion parameter model if your 50 billion parameter model has access to a perfectly structured domain architecture.
Exactly. It's about the tools, not just the raw brain power.
I do want to push on a major caveat that researchers themselves are very transparent about, though.
To compute cost.
Yes. Enabling this skill library, enforcing the DAG, running environment checks, it comes at a computational cost. The report notes that operating inside NeuroClaw increases overall token consumption and runtime.
Considerably, yes.
The models take longer to execute the tasks. So if it costs more compute and takes significantly longer, is it truly an upgrade in a world where speed is often the primary metric for AI?
It is a crucial trade-off to understand. Yes, the AI is thinking longer inside NeuroClaw, it is burning more tokens, But consider what it is doing with those tokens.
It's not just generating text.
Right. It's actively checking package dependencies. It's evaluating alternative tool chains. It's verifying that the dataset semantics actually align with the 𝗏𝗏𝗏 standard before it blindly commits to a calculation.
So it's performing the due diligence of a cautious human researcher.
Exactly. And the researchers proved the value of this extra compute using a metric called normalized gain.
Normalized gain. What exactly does that measure?
It measures the performance improvement relative to the theoretical maximum score, adjusted for the compute spent. The normalized gain numbers in the report clearly demonstrate that the extra tokens are doing real scientific work.
So it's worth the wait.
Absolutely. NeuroClaw forces the model to prioritize solution quality and scientific validity over raw speed.
Which brings us right back to the reproducibility crisis we started with. Rushing through a massive neuroimaging dataset and getting an invalid, unreplicable result doesn't actually save you time.
No, it corrupts the literature and sets the entire field back.
Right. In science, doing it right is vastly more important than doing it fast.
The rigor is the product.
So, synthesizing all of this for you, the listener, what does this all mean? NeuroClaw isn't just a new tool you install to make coding a little faster. It represents a shift from fragile manual pipelines to auditable, iterative experimental loops.
It is the automation of scientific rigor. But we do need to anchor this excitement in the present reality of medical research.
A reality check.
Exactly. The authors of the technical report are very deliberate in outlining the translational realities of this system. Strong benchmark performance means the system has achieved infrastructure readiness, not clinical readiness.
That distinction is incredibly important.
It is. Just because NeuroClaw can perfectly orchestrate a complex pre-processing pipeline on a clean, anonymized dataset like HCP young adult doesn't mean you deploy it unsupervised in a neurology ward to process live patient scans.
Because the stakes are entirely different.
Yes. We are dealing with highly sensitive patient health information. There are strict requirements for data governance, privacy compliance, and secure compute environments.
And I imagine human oversight is still non-negotiable.
Absolutely non-negotiable. Even with a perfectly deterministic pipeline, there is an absolute necessity for human supervision to catch AI biases and validate the technical outputs.
Right. An automated system can execute a pipeline flawlessly, but a human expert still needs to review those structured JSON audit logs to ensure the clinical context of the output actually makes sense.
The traceability provided by NeuroClaw is a tool for human accountability, not a replacement for human oversight.
The audit trail empowers the scientist. It doesn't replace them. It's a great grounding reality check. And as we wrap up this deep dive, I want to leave you with a final thought to mull over, building on the trajectory of everything we've explored today.
Let's hear it.
We started by highlighting the immense complexity of neuroimaging data, how murky it is compared to, a simple binary X-ray of a broken bone. Right now, NeuroClaw is designed to perfectly orchestrate and verify our existing scientific workflows. It runs our current software better. It enforces our current metadata standards flawlessly. But if these closed-loop agentic systems can synthesize multimodal data, manage computational environments, and iterate on multi-stage experiments with this level of deterministic precision, how long until they start proposing entirely new ways to process brain data?
Wow. That is the ultimate question, moving from execution to innovation.
Right, because if it understands the raw DICOM matrix so intimately and isn't bound by human cognitive bottlenecks, when does an agent like NeuroClaw stop being a brilliant lab assistant that just follows our established blueprints?
When does it start leading the research?
Exactly. When does it start discovering patterns or designing experimental modalities that human neuroscientists haven't even conceptualized yet?
I mean, once you successfully automate the foundation of reproducibility, the ceiling for discovery becomes limitless. The AI stops verifying the map and starts expanding the territory.
Something to keep in mind the next time you look at a brain scan and wonder what we might be missing. Thanks for taking this deep dive with us.